DNA is a molecule of two strands that form a double helix. Each strand is made of four chemical bases made up of the letter A, T, G, and C. Topoisomerase enzymes cut a strand, then bring it through the helix, then reseals it. The single strands are necessary because they keep the strands from colliding. The primer is a short strand of DNA, and it produces an enzyme called primase. Each DNA strand runs in the opposite direction. 3 to 5 inches is a leading strand, 5 to 3 inches is a lagging strand. An enzyme called polymerase binds the leading strand by adding a new ACGT to the lagging strand. A chunk of DNA called OCA-Z fragments are added. Finally, an enzyme called ligase joins together two strands of DNA. During mitosis, one cell divides once to form two identical cells. There are 5 phases in mitosis. The first one is interphase. During interphase, outside of the nucleus, there are 2 centrosomes, each containing a pair of centrioles. These structures are critical for the process of cell division. During prophase, the chromosomes turn into an X shape. Each chromosome is composed of 2 sister chromatids, each containing identical genetic information. During metaphase, the chromosomes line up end to end along the center of the cell. The centrioles are now at opposite ends of the cell, with the spindle fibers extending from them. During anaphase, the sister chromatids are then pulled apart by the spindle fiber, which pulls one chromatid to one end and the other chromatid to the opposite end. During cytokinesis, the single cell then pinches in the middle to form 2 separate daughter cells. Meiosis is a process where a single cell divides twice to produce 4 cells. These cells are sex cells. Meiosis 1 has 4 phases. Homologous chromosomes pair up in the process of crossing over. Spindle fibers connect and homologous pairs line up. Spindle fibers contract and split, and the homologous chromosomes move to the opposite poles. The cell divides to form 2 daughter cells. Meiosis 2 is divided in 4 phases. Centrosomes move to opposite poles. Spindle fibers from opposing centrosomes attach to chromosomes. Spindle fibers contract and separate the sister chromatids. Cells divide to form 4 daughter cells. Mitosis and meiosis use the same steps for cell division, like prophase, metaphase, anaphase, and telophase. I will refer to them as 1, 2, 3, and 4. First, in mitosis, 2 sister chromosomes are formed, and homologous chromosomes are crossing over in meiosis. In mitosis, double chromosomes line up, while in meiosis, chromosomes line up homologous pairs. 3 and 4 of mitosis, 2 identical daughter cells are formed. In meiosis, 2 daughter cells are formed. In phase 2, meiosis 2, 2 cells divide into 4 non-identical daughter cells.